The testes is divided up into these septa, okay? And within each septa, and it goes on and on and on, okay? Within each septa are 1 to 4 seminiferous tubules, okay? So here's what that means. And they're really microscopic, they're really small, and there's a whole heck of a lot of them. And this literally means if I were able to take every single seminiferous tubule out of you, both testicles, and line them up end to end, I could go all the way around football. That's how much surface area in seminiferous tubules and 2 testes in a human. That's a lot of seminiferous tubules. One of the reasons why you make so much sperm, because that's where they're being made. Okay? That's one of the reasons. Men are always making and releasing viable sperm from the seminiferous tubules. We'll talk about why that is in a moment. So just like everything else, you have to learn the conduit or the pathway. And the question on the exam would be, what is the path in which sperm has to travel? Okay? Seminiferous tubule to the recti testes, and all the recti testes is plumbing. And from the recti testes to the head of the epididymis, then the tail of the epididymis, and then the vas deferens, and we've already covered the rest. So, here is that order of plumbing that you can just memorize on your own. Okay, because we've been there, done that already. So, it's going to be on the exam somewhere. Okay? Okay. Okay, so what's in the nerve, basically? Well, parasympathetic, sympathetic nerve fibers for obvious reasons, blood vessel, lymphatics, that supplies the testes, and we've already talked about that. That's the spermatochord. Okay? Okay. So, here's the take-home message. In males, gonadotropin-releasing hormone, LH and testosterone, are released in pulsatile manner, occur every several hours. Follicle-stimulating hormone is released in a similar pulse of longer duration. Spermatozoa are produced by the testes by the process called spermiogenesis, which requires 5 to 9 weeks, depending upon the species, how long it takes to make the sperm. But keep in mind, you have enough of seminiferous tubules to go around the football field. Okay? So, spermatozoa are released constantly from the testes because you have so much seminiferous tubules. Spermatogenesis is a result of mitotic and meiotic divisions and concludes by the process of spermiogenesis. Spermiogenesis is a process whereby spherical spermatotids are converted to a spermatozoa and released from the seminiferous tubules on their way to the epididymis. Okay? So that's what we're going to talk about here. So here is the hours, the time in hours of the pulsatile secretions of gonadotropin-releasing hormone, which stimulates the release of LH and FSH in pulsatile manner as well. And in turn, these pulses stimulate pulsatile releases of testosterone in pulses. But this is the release. The actual concentration of testosterone in blood plasma is more like that. It's more flat in terms of the concentration. These are the different pulses of release. Okay? And you have these pulses as a result of negative feedback inhibition of testosterone at the level of the pituitary and the hypothalamus in the release of gonadotropin-releasing hormone and FSH and LH, right? You've been there, done that, right? So, here is a cartoon of Sertoli cells. So this is a Sertoli cell, this thing right here, okay? And these gaps in here are where spermatogonium was supposed to be. But this image leaves them out because we could care less about them on this image for now. So, the gonadotropes, LH and FSH, LH will stimulate luteinizing hormone, right? Will stimulate the Leydig cells. And the Leydig cells make testosterone. Testosterone ends up in your blood. Some testosterone gets shipped off to the Sertoli cells. So the counterpart for Leydig cells, and I'm just mentioning this in passing now, we'll come back to this. We're doing the female reproductive system. The counterpart in the female to Leydig cells are theca cells. Okay, theca cells. So you don't have to write that down. Just put it in your mind, and I'll reiterate that when we get to female reproduction. So the Leydig cells make testosterone. So in the female, the theca cells make testosterone. Okay. So then sends the testosterone over to the Sertoli cells, and the Sertoli cell aromatase activity. The Sertoli cells convert the testosterone into estrogen, and then that goes in your blood. So men are producing estrogen and testosterone. Okay? But more testosterone, of course. And the Sertoli cells also make what we call androgen binding protein, which binds up testosterone so that you have a high concentration of testosterone in the ejaculate for the maintenance of sperm development. Okay, in the epididymis in particular. Okay. So the Sertoli cells, under the influence of FSH, make a hormone called inhibin. Where's their inhibin? And inhibin feedback inhibits at the level of the pituitary the release of FSH and FSH alone. And that's not important in males. It is crucial in females, though. So we'll describe, or we'll expand upon that description when we get to that point in female reproduction. Okay? So, yeah. So in addition, the Sertoli cells have some really, really tight junctions between them. And they generate the blood-testis barrier. Because you don't want your immune cells to see sperm, because you'll mount an immunologic reaction against your sperm. Okay? You don't want that to happen. So your immune system really never sees the sperm because of the blood-testis barrier. And when they get into the epididymis, they're absorbed, so your immune system never sees them. Absorbed, broken down, and those products of sperm break down or recirculate.
Does sperm have a shelf life, not used?
Yeah, we can talk about that. Okay, so, a little story, okay? I used to, like I said, I used to teach at 2 universities, and during that experience, I used to have a urinary lab, where when we go to lab, I pass cups out to everybody in the class, and everybody in the class, men and women, have to go to the restroom and collect some pee, some urine. And then we take the urine, everybody would have to analyze their urine in lab. That was actually a lot of fun. Okay? So one day, I'm sitting there just watching my students, they were measuring potassium, they were measuring calcium, they were measuring sodium in the urine, and then they measuring nitrogenous waste products too, and then they'd have to look at it underneath the microscope to see if there was any proteins. I caught some urinary problems in this class with some of my students. So this one student came up, Dr. Gonzalez, she said, what are these? And she happened to set up her scope perfect, okay? So I looked in there and I said, whoa, check those out, okay? And I looked up like this, and I was going to tell her, but there was too many people listening. So I took her off, come here. Took her off to the side, and I said, those are sperm swimming in urine. And she says, I haven't had sex for 3 days. Well, I really didn't need to hear that, okay? But that's what she told me. And I believe her. So they used to think that the life expectancy of a sperm was 24 hours. Now they say it's 48 hours. Now, it might be somewhere on the order from 3 to 6 days as far as we know. Okay?
Without leaving the male, though. But just you lock them up.
No, you can potentially have viable sperm swimming in you for up to 3 days.
No, not me.
In the male, too.
The male.
Yes.
Okay, I didn't know if they got, recycled, if not used.
If they're in the prostate, if they're anywhere in the urethra or the ejaculatory duct, they're just hanging out.
Just waiting. But if nothing
They got
happens.
plenty of fructose to munch on. Okay? So now here's a story, and this was brought up in the last class too. So this woman had a baby. She was white, but she had an African baby. And her husband, who was white, said, been cheating on me. Well, as it turns out, he was cheating on her. He, and this is probably a true story, so he was having sex at the local prostitute shop, okay? And the prostitute he had sex with an African descent. And he has a foreskin, so some of his sperm stuck to his foreskin. And then when he had sex with his wife, he gave that guy's sperm to his wife and had a kid from somebody else.
That's disgusting.
Yeah, but that's basically how he had. So, but see now, in order to unequivocally prove that, they have to do genetic testing on the guy that had sex with the prostitute, and the prostitute has to admit that that's who he was, but she has to know him. But that's basically what happened. He was cheating on her. She wasn't cheating on him. Okay, so another strange phenomenon that is implemented by the abnormally lengthy life of a sperm, and how much insult that they can take. Hanging out on the foreskin.
Well, toilet seat babies.
Huh? And the woman in my class, where was her sperm? They were probably in her urethra, and she peed them out. So they were hanging out in her urethra for 3 days.
What about toilet seat babies?
No, it's not going to happen. Okay? Toilet seat babies. You got to be kidding. Question.
Okay, so why do they make you want to take Plan B in that 24-hour period?
Why do they want you what?
To take Plan B in that 24-hour period if sperm can still be in there up to 3 days?
Well, because what they're doing is they're stimulating you to have an abortion.
Yeah, but like they say, oh, it's too late after like 3 days, but it's...
Because that abortion's not going to work after the embryo gets into the uterus. See, is what you're going to do is you're going to stimulate. Plan B is generating a hostile environment in the uterus. If the fetus is already there, it's not going to happen. So what is that, the morning after pill? Is that what you're talking about? The morning after pill is stimulating an abortion. So you're pregnant. It's fertilized.
That quick?
Well, maybe not.
That's what I'm saying.
It depends. It depends. Now, if the egg is there already, it's going to take around 4 or 5 hours before fertilization takes place. Because the sperm have to get from the cervix all the way to the oviduct. That's going to take hours. Okay? Once that sperm's there and it fertilizes, then you go through phases called pre-implantation embryo development. In other words, the developing egg has to travel from the site of fertilization into the uterus. Once it gets in the uterus, the newly developing embryo has to signal the mom to maintain the uterine lining, and we'll talk about that later. If that signal occurs, then she's going to get pregnant. If the uterus is an inhospitable environment, it doesn't matter if the newly formed embryo signals or not. It's going to, because most of the time, people don't realize this, most of the time, the uterus is an inhospitable environment for embryo development. You have to grow the functionalis of the uterus and make it a hospitable environment for pregnancy to occur. So with the morning after pill, that's going to hinder that, and that environment will cause the egg not to implant, and that's it. It's a spontaneous, or you're stimulating a spontaneous abortion. But keep in mind, a lot of people don't realize this too, because the study was done that 40% of all pregnancies fail. 40% of all pregnancies do not go to term. Most of that 40% of pregnancies fail before implantation. Okay? That's a lot of pregnancies. That's a lot of embryos. So now keep in mind, this is the way researchers think and reproductive physiologists like myself think. You know, and so if 40% of all pregnancies fail, and most of those before implantation, that must mean, if there's a God, he's pro-abortion. So why are you aborting these fetuses? Because in all likelihood, there's something genetically wrong with it. If you're aborting a fetus and you're 2, 3, or 4 months pregnant, it's probably a good thing that happened, because there's something seriously genetically wrong with it, and 98.9% of the time, there's something wrong with it. Okay? If there's nothing genetically wrong with it, there's no reason why it will abort. Unless the woman fell down and hit her tummy real hard. That can stimulate a spontaneous abortion too. Or she's taken some drugs that can stimulate abortion, because that happens too. In fact, somebody in my family was trying to have an abortion because they were having a difficult marriage. She was taking some drugs from the Kudadeta, which is the local witch. Okay? And what happened to her kid? Well, she had the kid. But now it was born with tetralogy of Fallot, a severe heart condition. Now the kid doesn't know this even to today. But her mom gave her daughter a heart condition because of stuff she was taking during a crucial time of development that was involving the heart doing the twists. The heart develops as a tube, splits up into 2 tubes, and then it twists. And if it doesn't twist all the way during development, you're going to have cardiac anomalies. Her mom gave her those cardiac anomalies. And that happens too. Or you can be drinking too much alcohol at a certain time of pregnancy, or smoking too heavily, or having something else that you're exposed to during pregnancy. There's all kinds of things that can lead to abnormalities in fetal development, including neuronal abnormalities and even mental retardation. There's a lot of things that could be going on. So when you're pregnant, you need to guard yourself. Somebody smoking, get out of there. Because you want your fetus, your eventual baby, to reach a point where it can obtain its intellectual optimum. So you got to keep mommy happy when she's pregnant, because that helps. Okay? That's why I used to go, I'm your daddy. I'm in here. Can you hear me? And then, the kid will start kicking back. It was fun. And then we kick at each other like this. Of course, she found it annoying sometimes, but it was fun. And then when he was born, he recognized my voice when he was crying on the scale. Because I said, don't worry, okay, Daddy's here. And he just stopped crying, right, just like you turn off the switch. Then he raised his head like this, like he was looking. That was really cool. Okay. So, men, get involved in the pregnancy. Okay? You have to stimulate yourself to become attached. In women, it's automatic, and we'll talk about how that occurs hormonally a little later. Okay, we're done with this slide. Okay, so here's a cross-section of seminiferous tubules by scanning electron microscopy. Okay, so here is a seminiferous tubule, here's another one, here's another one, and here's another one. And keep in mind, these might be all the same tube that's like curling in and out of the plane of focus. Or it could be up to 3 or 4 tubes, but they're all doing the same thing. So when you cut it, you're going to get morphology like this. Okay, so now I want you to look at this, and then I'm going to describe what's going on here. Now note that in the seminiferous tubules, because this is two different images, because you can tell they're different colors, so it's two different images. So look at the lumen of this seminiferous tubule and notice the lumen of this seminiferous tubule. They look different. Notice this seminiferous tubule as compared to the other two I just circled. It looks different too. Okay? And here's another one. Check that out. So big differences no matter where you look. So each one of these is a cross-section of a different segment of the seminiferous tubule, and each one looks different. Okay? That's an important concept that we're going to come back to. So here's another seminiferous tubule, another section, and I have a slide like this in the lab someplace, and these look different too, but it's not as apparent as the previous one because the previous was a better prepared slide. Here is the epididymis, and I think this is human, and all this stuff right here is a lot of sperm. Okay? And then you have the seminiferous, again, it's a tube that goes in and out of the plane of focus. It's just one tube. Okay? So it's wrapping all over the place. So here is, and I've seen this slide today too. Here you have seminiferous tubules on this side, epididymis on this side. Okay? Separated by tunica albuginea or dense irregular connective tissue. And here's the spermatozoids. Okay, since I'm here, how about another story? Okay? Okay, you like the story? Okay, so I'm teaching at Arizona State University again. And, I used to run some labs. And one of the labs I used to like to run, I like to get some volunteers from the audience to give me some sperm. So I said, I will give you a culture, some culture medium in a jar, and you can take it home and bring me some sperm tomorrow or the next time before we meet for class. And every once in a while, I get a guy to donate, okay? One time, one guy said, well, why don't you do it? I said, I can't. Okay? I was vasectomized a long time ago, so I couldn't produce. So I said, any volunteers? I said, I'll even give you extra credit. So that was the bite. And then the one time, I didn't even offer extra credit, and this woman in the front said, I'll do it, Dr. Gonzalez. I'll get you some sperm. I said, okay, and I didn't question her how she was going to do it. I just gave her the medium, okay? So she brings me the cup, the day before lab, or the day of lab. So we go in there, and I had this real cool setup, okay? I have an inverted microscope. So I take some of the sperm, I swirl it around, and I put it in a petri dish. I put the petri dish on an inverted microscope because the objectives are looking up, not down. So you can look right into the petri dish. And my scope has a camera on it, and the camera is attached to a computer screen about that big, and another video screen about half the size of this. So, most of my students, I had several groups of students get to look at the sperm, right? And she's there, she's watching, right? She's like over my shoulders, and she's like getting in the way. I said, and she's there because, it's her sperm, right? And I didn't tell her, I didn't ask her how she got it, but, it was her boyfriend's sperm. And all of his sperm looked like that. Every single one of them had a bend in the cilia. Okay, now earlier I said, the tail for the sperm is worthless until it's in the vicinity of the egg. When it's in the vicinity of the egg, it has to swim straight. These little guys were swimming in circles. They ain't gonna fertilize nothing. Okay? So I said that, I said, look, there's a problem here. She says, what is it? All over me, like, you would not believe. I had to go like this to get her away a little bit. Okay? I said, because these swim in circles, and I explained to her why that's not going to do anything, right? Not going to fertilize, because they got to swim straight through that cloud of granulosa cells and get to the zona and get in there, because they got to swim straight. These can't swim straight. And she said, I knew it was him.
She was doing research.
They were trying to get pregnant, and she couldn't get pregnant, and he was blaming her. Okay? And as it turns out, it was him. So I said, damn, I should charge you $500 for this. This is a freebie. So I hit a button and printed off a color copy for her. And I said, here, give this to your doctor and tell him what I said. Okay? So what would she have to do? She would have to get in vitro fertilization. She can still get pregnant from him. They have to take his sperm and inject it into the egg. Intentionally. Question?
My sister-in-law had to get him in vitro twice now because they said he had flathead sperm. I guess like a hammer shark, I don't know.
We'll talk about the acrosome later, and that's probably the problem he was having. So you have to undergo the acrosome reaction. We'll deal with that next time, okay? So, no kidding. This woman was cunning. She got really a freebie, okay? She saved some money, okay? Which I thought was pretty slick, so I let it go. I didn't charge her anything for that. So, okay, let's get back. Okay, so let's explain the differing morphologies in the seminiferous tubules. So what's happening in the seminiferous tubules? Well, meiosis is happening. You have the spermatogonium at the base, so this will be the connective tissue, and this will be one big Sertoli cell making up seminiferous tubule. And the sperm go through stages to spermiation. Okay? So some guy just sat around looking at different sections of seminiferous tubules, and he got this information. So what he did is he looked at every seminiferous tubule, and then he started categorizing sperm development in every tubule. Meant that, I mean, this was really kind of done back then, but the guy didn't have a life. And he got this data published, and this is out of one of his publications. Okay, so, and I'm not going to, don't bother memorizing all this, just know that, that's what he did. So after the sperm leave the seminiferous tubule, they look like this. Okay? And then when they end up in the ejaculate, they look like this. So you have to undergo epididymal maturation. So there's different stages of development. So here's your seminiferous tubule, and here's the different stages of development, Roman numerals assigned. And in some areas of the seminiferous tubules, you get almost complete development. In others, you don't have much of anything. In other words, different stages of development, and each section of the seminiferous tubule is within each section, its sperm development is synchronized. Outside of that section, it's not in synchrony. And remember, you have seminiferous tubules that can go all the way around a football field, which means you're cranking out viable sperm every second of the day. Okay? They're just coming out. Okay, so that's interesting anyway, and that's why different cross sections of seminiferous tubules look different. High magnification of the oviduct, and you can see cilia in these folds in the oviduct. Okay, these cilia. This is really good technique that this histologist did because these cilia are very fragile, come off really easy. There's higher power of the cilia. And these are ciliated simple columnar epithelium, not like your respiratory tract where they're ciliated sort of stratified. These are ciliated simple columnar. And here's the oviduct, okay? They're high magnification of the same slide, okay? And just for comparison, here's the respiratory tract, there's cilia, okay? Let's get back to the lecture. So you have cilia in the isthmus and cilia in the ampulla. What are these cilia doing? And there's also smooth muscle in the oviduct, surrounding the oviduct. So you got peristaltic contractions of the oviduct, and the peristalsis is moving like this, and it meets in the middle. So the peristalsis is pulling the egg from the ovary and pulling the sperm from the uterus to the ampulla-isthmus junction. In other words, peristalsis is in opposite directions to the point of the ampulla-isthmus junction. In addition, the cilia are doing this in opposite directions. That's weird. Cilia are doing this. Peristaltic waves are doing this. They meet in the middle, and right after fertilization, peristalsis reverses. Cilia do this now. Peristalsis does that. It reverses in the isthmus, taking the newly fertilized egg in the opposite direction that the sperm came. The sperm didn't get there with those little tails. Those tails are worthless until the sperm is in the vicinity of the egg. Then they serve a purpose. Because getting from the cervix to the ampulla isthmus junction, where fertilization takes place, will be like throwing me in the Atlantic Ocean and telling me to swim to Japan. I ain't gonna make it. Okay? Ain't gonna happen. Okay? So how do the sperm get there? Peristaltic contractions of the uterus, peristaltic contractions of the oviduct, and ciliary activity by the oviduct. The sperm be. Then when they're in the vicinity and they're touching or near the egg, then that tail gets them through that granulosa cells, the zona pellucida, finally to the egg. That's when the sperm tail is important. Okay? Other than that, otherwise that sperm's tail is pretty useless. So fertilization takes place here, and there has to be some mediator released by the newly fertilized single-cell embryo. And by the way, it doesn't stay a single cell very long. 4 hours after fertilization, it's two cells. Okay? So on day one, it's a 2-cell embryo. Okay, we'll talk more about that a little bit later, but some mediator has to be released by the early embryo that tells the cilia and the smooth muscle of the oviduct to reverse peristalsis and ciliary activity to get that egg out of the oviduct and into the uterus. Because it ain't going to get there on its own either. We call this the first recognition of pregnancy. You don't know you're pregnant, but your reproductive system does. Called the first recognition of pregnancy. That means there's a second. We'll talk about that one later when it's appropriate. Okay? So I told you there's some really cool stuff going on. You can see that the cross sections of the ampulla and isthmus are slightly. Okay. So sperm arrive in the ampulla isthmus junction. Cumulus release sperm motility factors, the cumulus or the specialized granulosa cells. Sperm become hyperactive. The acrosome reaction takes place. And then, in other words, you're releasing all those enzymes. And then you get penetration of the cumulus and the corona radiata, which are the specialized granulosa cells that we'll talk about later. And then you get binding of the zona pellucida, penetration of the zona pellucida, sperm-egg membrane fusion, sperm engulfment, decondensation of the sperm nucleus, formation of the male pronucleus, and in sperm oocyte membrane fusion. Okay, the acrosome reaction is an excytotic event that allows cells to penetrate the zona pellucida and fuse with the oocyte. We'll talk about this a little more. So here's the acrosome reaction in our artistic rendition. We have the dark area right here is the sperm nucleus, and this would be acrosomal contents or the acrosome cap. You get the acrosome reaction, and the enzymes get out. Hyaluronidase, as mentioned, esterase, procretin gets converted to acrosin, acid hydrolase breaks everything else down. Sperm motility factor from the cumulus makes them hyperactive. Hyaluronidase disperses the cumulus cells. Esterase breaks down the sperm radiata. Okay, we'll talk about these later, so you've got to come back. So hyaluronidase is an enzyme that breaks down the hyaluronic acid. Hyaluronic acid is a glucosaminoglycan. Remember from last semester, the GAGs? Glucosaminoglycans? We talked about that. Did you talk about glucosaminoglycans in your class? Glucosaminoglycans. You don't have to make a lot of melon. That's what's happening. So So glucosaminoglycans are part of the connective tissue in your skin, and the glucosaminoglycans hold the cumulus saloperis and the corona radiata together. Okay? So you need hyaluronidase to get through the hyaluronic acid, which is hyaluronidase is an enzyme that breaks down hyaluronic acid, glucosaminoglycan, which is part of the connective tissue holding the cumulus cells together around the egg. When the sperm makes contact with the cumulus cells, it encounters a sperm motility factor produced by the granulosa cells. The sperm becomes hypermotile at this time for the penetration of the cumulus on the way to the zone of the cell. So these specialized granulosa cells, called the cumulus saloperis, that you ovulate with, your egg, are there to provide protection to the newly ovulated egg and to provide sperm motility factor to assist the sperm doing its thing. Corona radiata, which are specialized granulosa cells that are attached to the zona pellucida, do something else, and that's really cool too. Okay, we'll talk about that later.